Okay, good morning, everyone. A very warm welcome to all our participants joining us today, both from University Technology Mara and our guests from abroad. My name is Associate Professor Dr. Yong Soon Kong from the School of Chemistry and Environment here at FSG UITM, Shah Alam, and I am honored to serve as your moderator for today's session. This morning's international invited lecture is organized as part of the Soil Science and Analysis course. We are delighted to explore a topic that is becoming increasingly vital, and this is soil phytoremediation, how plants heal contaminated soil. Ladies and gentlemen, soil contamination remains a global challenge, but nature often provides its own solution. Today, we are privileged to learn from an expert about how we can harness the power of plants to recover and heal our environment. It is now my great pleasure to introduce our distinguished guest speaker. Joining us from Universitas Indonesia, please welcome Associate Prof. Dr. Ratna is a of biology within the Faculty of Mathematics. Her contribution. Further ado, I invite Dr. Ratna YUNIATI to stage is yours.
Okay. Very good morning, everyone. Allow me to introduce myself. I'm Ratna YUNIATI from Department of Biology, Faculty of Mathematics and Natural Sciences, Universitas Indonesia. Today I will deliver a topic, soil phytoremediation and how plants heal contaminated soil. Before we go into the lecture, I'd like to start with simple questions. Can plants really clean polluted soils? Well, maybe it may sound surprising, but plants are not just passive victims of pollution. Many plant species actively interact with contaminants in the soil and working together with microorganisms to reduce environmental damage. And this is the idea of the basis of phytoremediation. And today we'll explore how plants can become natural helpers in restoring contaminated soils. Nowadays, many soils are actually sick. They contain substances that should not be there, such as heavy metals, industrial chemicals, microplastic, and other pollutants. And as shown in these diagrams, contaminated in soil come from many sources like volcanic eruptions, forest fire, vehicle emissions, and intensive agriculture and urbanization. And contaminants can move from soil into the plants, uptake by root or uptake by foliar. And this can be affect plant growth, physiological process like photosynthesis, and overall ecosystem stability. And when soil is sick, it cannot function properly. It cannot support healthy plant growth or sustain a balanced ecosystem. So just like humans, soil also needs treatments. And this treatment is called soil phytoremediation. In this slide, I want to change the way we usually think about plants. We often see plants as passive victims of pollutions. They get stressed, damaged, or even die. But in reality, Many plants are much more active than that. As you can see in this illustration, plants interact continuously with contaminated soil. This soil is contaminated by pollutants. So plants, through their roots, they can take up the pollutants, translocate it to the leaf and immobilize them. So plants are not just affected by pollution, they can actually become part of the solution. This is why we say plants can as active agents, helping soils recover rather than simply surviving in polluted environment. And so what do we mean by phytoremediation? The word comes from phyto, meaning plant, and remedium is restoring balance. So the idea is very simple. It's the use of plants to accumulate, to remove, transform, or detoxify pollutants from contaminated soil or water. So what should plants do in phytoremediation? Basically, plants can respond in four main ways. First, plants can take up contaminants from soil into their roots. Second, plants can bind or hold the contaminants inside their roots tissue, either physically or chemically. And third, some plants can transport contaminants from their roots to the shoots. And finally, plants can also prevent contaminants from leaching out of the soil so they do not spread to other areas. This slide explains what we discussed in the previous slide. It shows how plants take up and manage heavy metal inside their body. First, heavy metal, this symbolized by this circle in the rhizosphere. They enter root cells and can be stored in the vacuole. This is a uptake pathway in roots. And also, the heavy metals can transport it upwards through the xylem from roots and to the shoots and leaves. And in the leaves, plant can store safely in vacuole. This is a vacuole in the cell leaf. So plants do not simply absorb heavy metal, but they actively manage where the metals go and how they are stored. So if the heavy metals already sequestered or isolate in the vacuole, they cannot harm the cell. And at this point, a common question is why plants can survive contaminated soils. So one important reason is the root systems. Roots are not passive pipes, but they are active organs that explore the soils. Root can selectively take up water and nutrients and at the same time limit the entry of harmful substance. Roots also release substance called root exudate, and this exudate can change soil chemistry such as pH and redox condition. And by changing soil chemistry, roots can influence metal solubility, making some heavy metals less available and less toxic. So roots help plants adapt to contaminated soil. Secondly, why plants can survive contaminated soil is the physiological plasticity. This means that plants can adjust their growth and physiology in response to environmental stress. You can see this plant in normal soil, that's soil not contaminated by microplastic or nanoplastic, like in this contaminated soil. These plants is growth in normal condition. They emit flower. This is fruits. And while at the contaminated soil, the plants is maybe rather stunting because they not prioritize the maximum growth. They just look different, but they are still able to survive through adaptive response. They modify the root function, growth pattern, and physiological response to cope with soil contamination. So this is adaptive response to soil contamination. Another key reason why plants can survive contaminated soil is plants do not grow alone in the soil. Instead, they make a symbiosis with microbes. So as shown here is the rhizosphere is a very active zone where roots and microbes interact continuously. These microbes help plants cope with environmental stress. Some microbes can change the chemical form of the contaminants, making, let's say, heavy metals less toxic or less available. And other microbes help improve nutrient availability and support plant growth, even under contaminated condition. So through this partnership plant microbe, plants gain extra support to survive and function in polluted soil. And then who helps the plants? So in the rhizosphere, plants interact with different group of beneficial microbes, including bacteria and fungi. Bacteria is, this example is plant growth promoting rhizobacteria, and the fungi is mycorrhiza, AM fungi or EM fungi. This microbial community support plant growth, nutrient cycling, and help reduce stress which caused by contaminants. So the key message here is that plants never walk alone. Now let's look at the key mechanism behind phytoremediation. Phytoremediation does not rely on a single process. Instead, it involves several different mechanisms. You can see some plants can extract the contaminants which is in the rhizosphere from the soil, translocate to the leaf, and store the contaminant in their tissue. And this mechanism is known as phytoextraction. In other case, plants can immobilize contaminant in the soil, preventing them from spreading. And this called is phytostabilization. In phytostimulation, plants enhance microbial activity in the rhizosphere and allowing microbes to degrade contaminant. Just like in phytodegradation, this pollutant, and by enzyme, they degrade the contaminant. And finally, plants can reduce contaminants by volatilizing certain compounds or by filtering pollutants from soil. So the important point is that different plants and different sites may involve different mechanisms. And how plants heal contaminated soil? First is uptake and removal of contaminants. So this figure illustrates how plants take up contaminant from the soil, and the contaminant enters the plant through roots and then transported upwards to different plant tissue like leaf. Some of this contaminant can be stored inside a plant cell, reducing their concentration in the soils. So plants function as natural filters, removing pollutant from contaminated environment. In this second mechanism is transformation of contaminants. Plant and soil microbes can change contaminants. Plants do not just absorb contaminant, but they actively transform the contaminant. Together with soil microbe, plant roots release enzyme and exudate. This exudate can make a complex with heavy metal. So the heavy metals is not toxic, but converted into less toxic and less mobile or more stable form in the soils. So here the soil is healed not by removal but by chemical and biological transformation. So this highlight that phytoremediation is not passive uptake but an active biochemical process involving plants and microbes. The third way plants help heal contaminated soil is by improving the soil itself. Plants' roots change the soil structure by creating pores and aggregates so water can infiltrate better and roots can grow deeper. As you can see in this picture, this is a good soil structure. The root can grow deeper compared to the plant which grow in the poor soil structure. There are some surface crusting that limited root development. So better soil structure can reduce the bioavailability of contaminants, making them less accessible to plants and soil organisms. So here, healing means restoring soil functions, not just removing pollutants. This soil improvement explains why phytoremediation works as a long-term restoration strategy. The fourth way plants help heal contaminated soil is by stimulating soil microbes. Plant roots release compound into the soil, like in this picture, is a root exudate. It could be sugar or organic acid or amino acid. And this compound support beneficial microbes, which bacteria and fungi. And microbes will help degrade or immobilize contaminants. So in this process, plants do not work alone, but cooperate with soil microbes to support soil recovery. Sorry. And the last way how plants help heal contaminated soil is prevention of contamination spread. These plants reduce soil erosions and surface runoff, and leaves is the canopy is intercept rainfall, and roots improve soil structure and water infiltration. And as a result, contaminants are less likely to be carried away by water or wind. Or just in simple terms, plants help keep pollutant in place so they do not spread to the river, groundwater, or surrounding area. At a broader ecological scale, vegetation helps stabilize contaminant within the soil system. So the plant prevents contaminant spreads. Instead of moving freely, metals tend to remain bound in the soil, stabilized in the soil. Vegetation is reduce wind-blown dust, limit erosions, and slow down surface runoff. And this stabilization improve ecological function and reduce risk to surrounding ecosystem. So here, plants act as ecological barriers rather than just physical cover. After understanding how plants can heal contaminated soil, the next question is what types of plants are actually used? In phytoremediation, different types of vegetation can be selected depending on the contaminant and the site conditions. Grasses, like you see in this list, are often used because they grow fast and have extensive root system. It's important in phytoremediation is we choose the plants who grow fast and have a big big biomass, so they can take up and can store the contaminant in the biomass. Legumes such as alfalfa are useful because they improve soil quality, because legumes have a symbiotic relationship with rhizobium. They can fix the nitrogen from the atmosphere and they can help plant get nitrogen. And trees like poplar are effective for deeper contamination. Some plants such as sunflower, Indian mustard, and vetiver grass are well known for their ability to tolerate and accumulate heavy metals. So plant selection is a key factor in successful phytoremediation. One example of terrestrial plant used in phytoremediation is alfalfa. You can see this, its flower, leaves, and the fruits. Alfalfa has a deep and extensive root system which allow it to explore contaminated soil effectively. Through a root system, alfalfa can absorb and tolerate various pollutants, including heavy metals and polycyclic aromatic hydrocarbons, is organic pollutants. And most of these toxic elements are retained in the roots, which help stabilize contaminant and reduce their mobility in the soil. Another important group of terrestrial plant used in phytoremediation is poplar tree or populus. Poplar are fast-growing trees and have deep root system. As you can see, this poplar tree is maybe it's 10-meter high, but I don't know in the Malaysia, but in Indonesia they're not poplar tree. It's a subtropic tree. They also have transpiration, have high transpiration rates. This which help draw contaminants from soil and groundwater. And because of these traits, poplar are widely used to remediate heavy metals and organic pollutants. Poplar have many species within the genus, which has been studied, but their functional traits are more important than the exact species name. This is the example of how poplar is used in phytoremediation in the site. The plant is grow in a row like this, and if we zoom in this area, we can see this one or individual poplar trees. This is a tree trunk, and this is iron deposit, and this salt deposits. The deposit is left behind and accumulate near the root zones. And this process limit contaminant movement and demonstrate phytostabilization. And so here, the poplar help manage the contamination without necessarily removing all pollutants from the soils. Another example of terrestrial plant use in soil phytoremediation is sunflower or Helianthus annus. Sunflower is also fast-growing plants with high biomass production. It can take up several heavy metals such as lead, cadmium, and zinc from contaminated soil. As shown in the diagram, it's a research from Wang et al. 2023. Metals, which is in the soil as the substrates, can be taken up by roots and distributed to different plant parts, such as roots, stem, leaf, and maybe at the seed also. And because of these characters, sunflower commonly used as a model and also as a phytoremediation agent, and its effectiveness strongly depends on soil condition and contaminant availability. The next important terrestrial plant used in soil phytoremediation is vetiver grass or Chrysopogon zizanioides. Vetiver also has an extremely deep and dense root system and tolerant to metals and acidity, and this makes vetiver very effective in stabilizing contaminated soils, especially in area affected by acid mine drainage, like this illustration here. Most contaminants are retained in the root zones, which help reduce metal mobility and prevent further spread to wider area. And because of this trait, vetiver is widely used for soil stabilization and phytostabilization rather than metal extraction. This is a picture from I searched in Google. This because the vetiver is tolerant to heavy metal and can make a soil stabilization, erosion control, and groundwater retention. You can see the proportion of the roots is much bigger than the shoots. Another terrestrial plant is sorghum sudanense. Sudan maybe is from Sudan area. Maybe this indigenous from Sudan. This Sudan grass is a fast-growing species with high biomass production. So this character is important for phytoremediation. It's fast-growing and high biomass production. It can accumulate several heavy metals, including lead, nickel, and cadmium. And in some cases, a higher metal concentration are observed in the shoots than in the roots. So this suggests its potential for phytoextraction, especially when combined with proper soil and harvest management. However, its performance depends strongly on site condition and metal availability. Another terrestrial plant used in soil phytoremediation is red fescue or Festuca rubra. Rubra is mean red. They have extensive root system and high stress tolerance. And because of this trait, it's suitable for eco-restoration, land restoration, and they can accumulate several elements, including arsenic, boron, copper, and also from fly ash. Fly ash, it can be filtered also by the Festuca rubra. Another terrestrial grass used in soil phytoremediation is ryegrass or Lolium peren or perennial ryegrass. This species is known for its tolerance to petroleum contaminated soil, including crude oil. It can remove hydrocarbons as well as several heavy metal. And some study very high removal efficiency has been reported, especially for copper. And because of its fast growth and tolerance, lolium is often used in contaminated agricultural and industrial soils. Another terrestrial grass used in soil phytoremediation is Bermuda grass, Cynodon. And Bermuda grass has been studied for its phytoextraction potentials, particularly for copper contaminated soils. For example, this is a picture from studies from the Katanga region in Zaire. They show effective uptake for copper. And it has also been investigated for arsenic and mercury accumulation. And in addition, Bermuda grass perform well in petroleum contaminated sites, so making it a good choice species for different types of soil pollution. Beside the phytoremediators, there's also hyperaccumulator species. And what is a hyperaccumulator species? Hyperaccumulator species is plants that accumulate exceptionally high metal concentration. Maybe the metal content far exceeds normal physiological levels. As example, some plants from hyperaccumulator species can store nickel more than 1,000 milligram in every kilogram dry weights. So this is maybe for non-hyperaccumulator species, the concentration in this high will kill or make the plants into the toxicity condition. So why does phytoremediation matter? If we compare between conventional remediation and phytoremediations, the conventional remediation is expensive because have used the excavator to remove the contaminated soil, and sometimes they can destructive to the soil structure. And the conventional remediation is just short-term fix because maybe the pollutant can be back again to the sites. But in the phytoremediation is cost-effective because plants is just need sunlight and the carbon dioxide, and this phytoremediation is also eco-friendly, and it's long-term restoration. And this makes phytoremediation particularly valuable when long-term soil health is the main goal. And what is the benefits of phytoremediations? Phytoremediation offers several important benefits compared to conventional remediation methods. First is sustainable approach that works with natural process. Secondly, it requires relatively low energy input. Yeah, as I said before, because plants just use sunlight and natural growth process. And phytoremediation also provide aesthetic and ecological value by improving the landscape while restoring soil quality. You can see this is a picture taken before and after the phytoremediation is done. And finally, because of its lower cost and simpler technology, phytoremediation is especially suitable for developing countries. Oh, sorry. And phytoremediation is aligned with several sustainable development goals, especially SDGs 15, Life on Land, SDGs 13, Climate Action, and SDGs 6, Clean Water and Soil Health, by offering a nature-based solution for soil and environmental restoration. So phytoremediation connect plant science with global sustainability effort. And at this point, it is important to be realistic, I think. Phytoremediation is not a quick solution. Unlike conventional remediation methods, phytoremediation work as long-term process. The time needed depend on several factors, time and number of plant use. There's a type of plants which how they extract the contaminants, how it can translocate to shoots or just in the roots. It will make the phytoremediations fast or slow. And the time depends also on type and amount of harmful chemical presence, and size and depth of polluted area. Sometimes the polluted area is just in the surface, but sometimes it go deeper into the soils. And the last is type of soil and condition presents. However, this long time frame also allow the soil system to recover naturally. So phytoremediation is not a short-term fix, but a long-term restoration strategy. And to conclude, healing contaminated soil is not a purely technical process. It is a biological and ecological process driven by interaction between plants, microbes, and the soil environment. And although phytoremediation is not a short-term solution, it offers sustainable and nature-based approach for long-term soil restorations. And understanding how plants survive, adapt, and interact with contaminated soils help us to see plants not only a victim of pollutions, but also as active partners in environmental recovery. So the healing contaminated soil is a biological process and driven by not only plant, but also microbes. Thank you very much for your attention. And with that, I would like to return the session to the moderator, Dr. Yong.
All right. Thank you, Dr. Ratna, for the incredibly insightful and enlightening presentation. It is fascinating to see the mechanism by which plant species can act as natural cleaners for our soil. Now, I'm particularly happy with your presentation because you actually have indirectly make a revision with my students based on what you have presented. The soil physical, chemical, and biological properties, they're all revised within your talk. So thank you very much. Now, reason because in 1 month's time, my students will sit for final examination. So this is a good revision with you. Okay?
Yes.
Anyway, so we now move on to the next session, which is the question and answer session.
Yes.
So for those of the audience in Google Meet who wish to ask questions, so feel free to type your question in the chat box. Alternatively, you may use the raise hand feature in which I will call you upon to unmute and ask your question verbally to Dr. Ratna. So with that, so we welcome. Is there any question from the audience? Oh, wait a minute. I think we have a few questions, two of them in the chat.
Oh, yeah.
Starting with Puan Nur Firdaus Abdul Rashid, which is my colleague in UITM. She has a question for you. Is there any method to discard the plants? I assume that the plant's biomass after phytoremediation that has been restored heavy metals from contaminated soil. So perhaps, Dr. Ratna, you would like to answer this question first?
Okay, thank you. Ibu, I think Ibu or Bapak Nur Firdaus Abdul Rashid. Thank you for the question. Oh, Ibu, okay. Is there any method to discard the plants that have restored heavy metal from contaminated soil? Actually, if the contaminant already absorbed or binds in the biomass, we do not have to discard. They will senescence, growing old, the biomass, and the biomass will fall from the canopy to the soil, and the heavy metal is not toxic anymore or harm the soil because it's already fixed or already bind, and they cannot move or harm the soil anymore because it's already keep in the vacuole of root cell or leaf cell, so they cannot toxic anymore. So we don't have to discard the biomass. So the plants is just grow, extract, and store the contaminant in their cell. And when they fall down to the soil, they will just degrade and mingle or with the soil. So we just don't have to do anything. Just plants like usual plants. If the become litter in the soil, they cannot harm to the soil anymore.
Okay, thank you, Dr. Ratna.
Yes.
I think I will go on to the second question, in which I will revise the question, because I think your earlier answer perhaps have answered half of the question from the second audience, Nur Damia. Because at the end of her question, she asked, we just let the plants grow on their own. So the answer is yes, you have already answered. But perhaps the first part that needs perhaps your explanation. So her question: Do we not have to add anything to enhance phytoremediation? So what would be your response, Dr. Ratna?
Okay, there's phytoremediation, assisted phytoremediation. For example, the first is we inoculate the plants with the microbes. Example is rhizobium, the symbiosis. The microbes or bacteria or the fungi is just help absorb the contaminants, or another bacteria not help the how to degrade or how to extract the contaminant, but they make the plants grow better because they help how plants get the water, get the nutrient from the soil, and because the help from the microbes, the plants grow faster, grow better. Then the plants can extract the contaminant better. That's
Okay.
my answer.
Thank you, Dr. Ratna. Now, I think Damia is my student. So, if I can add, Dr. Ratna, if I can add
Yeah.
from the
Yes.
chemist's point of view. So, as a chemist,
Yeah.
I do a lot of chemical phytoremediation, chemical assisted phytoremediation. So, to
Yeah.
answer Damia on what to add, number one is fertilizer.
Okay. Yeah.
Yeah. So if I can add fertilizer, because some of the fertilizer that contains phosphate help chemically immobilize the heavy metals. Yeah.
Yes.
So that is the addition from me. Now I move on to the question from my good friend Dr. Lily Shahani, who also have similar expertise as yours. So we hope that later we can have further discussion on further collaboration. So Dr. Lily asked, can you discuss ecological risks associated with using fast-growing plants for soil remediation? So, Dr. Ratna?
Yes. Thank you for the questions. And from my point of view, using fast-growing plants for soil remediation can be effective, but I think it also involves several ecological risks. One major risk is invasiveness. If we falsely choose the species, fast-growing species may spread beyond the remediation site and can compete with native plants. That's cause a loss of local biodiversity. And another concern is food web transfer. If these plants accumulate contaminants, herbivore may consume them, allowing pollutants to move into the food chains. And fast-growing plants can also alter soil properties. They may change nutrient availability, microbial community, and or maybe water balance, which can affect surrounding ecosystem. And therefore, while fast-growing plants are useful for remediation, their use must be carefully managed to minimize the ecological risk. That's my answer.
Thank you, Dr. Ratna. Exactly what I thought of. Malaysia have fair share of experience importing non-native plant species, a tree species in fact, in Malaysia.
Yeah.
Yeah. We have imported one called acacia. I think acacia is also native to Indonesia, but
Yeah,
not,
yeah.
isn't it?
Mhm.
So I think so far we have three questions, and I would like to ask Dr. Ratna myself from the legal standpoint.
Yes.
I think each country have different regulation terms of in terms of using plant species for
Yes.
phytoremediation. I think this one is good for us to tell our students what are the legal framework. Like for example, some of the plant species they are native and endemic to Indonesia. So my question to you is, what are the challenges of what are the challenges for us to uh, realize the potential of these endemic hyper accumulators, particularly from Indonesia? Can they be exported to other countries to be used as as a plant species for their phytoremediation outside of Indonesia? So those are the questions, Dr. Ratna.
Okay. From legal point of view, soil remediation using plants, I don't know until now is maybe it's regulated differently in each country depending on national environmental policy. And when we consider the use of endemic hyperaccumulator plants, several challenges arise. First, many endemic species are protected by law, and which limit their collection, cultivation, and large-scale use. And second, there is often a lack of clear legal guidelines that allow the use of endemic plants specifically for remediation purpose. And as a result, their potential cannot be fully realized. Um, in Indonesia, I think in Malaysia also, there is one species which I know is Melastoma malabatricum. Is in local name is senduduk, I think of. Uh, this melastoma is have a good character is they can grow even in good condition in low pH in acid soil. And my supervisor already have a team to conduct the research. This melastoma can grow in aluminum concentration with high concentration of aluminum. So another research show that melastoma is all can extract the heavy metals such as lead, cadmium, cuprum, and zinc also, and that make the melastoma as one of candidate for phytoremediation. And I can also meet or found these plants in the area which have soil is not fertile as the marginal land. So melastoma can be used for phytoremediation in land that contaminated with heavy metal.
Okay.
That's my answer, Dr. Yung.
Thank you, Dr. Ratna. I believe that both Indonesia and Malaysia, and if I can add Philippines also, we are very,
Philippines.
we are very rich in biodiversity. That is why several unique plant species were found in Southeast Asian region. One of which you already mentioned is melastoma, and there are many more that are only found in Borneo, in Kalimantan.
Yes.
Yes. So, which is why these unique plant species, they are also our treasure, biological treasure, we call it. So that is why I think Indonesia and similarly Malaysian government has banned the exportation of these endemic species, even though they are very good in hyper accumulating metals from the soil. So I would say this is the knowledge for my students if they would like to know. So I think we probably can allow one more last question, which is from my student, Alif Hilmi, who has raised a hand. Alif, are you still here? If you're still here, you can unmute.
I'm still here, doctor.
Yeah. So go ahead. What is your question?
These phytoremediation plants, they either hyperaccumulate heavy metals or immobilize them in the soil, correct?
Yes.
How feasible is it to harvest these plants and use it to recycle the metals for consumption in the industry?
Oh yeah. It's people frequently call this phytomining. Yeah, because the heavy metal is absorbed by the roots and the plants translocate to the biomass, in the leaf. And if we can, if we want to extract the heavy metal from the leaf, is as theoretically we can harvest the leaf and crush or grind the leaf, and use the chemical reaction to withdraw the heavy metal from the tissue of the leaf. I think it's the theoretical, but I don't know if how the methods the methods to get the heavy metal or the metal from the plant's tissue.
Okay.
Okay. All right.
Thank you for
Okay.
the
Okay, Alif.
questions.
Thank you,
Yeah.
Dr. Ratna. I think I can tell Alif a little bit of
Mhm.
reusing, because that is also in Sabah, where project is also have the collaboration of a team from UITM Sabah as well. So there's one site in Mamut where it used to be mining copper, but not anymore. That site still have nickel. So they are planting hypo accumulators, like you said, phytomining. They are phytomining nickel from those soil. So to answer Alif, this is also one part of my project in which I reuse those biomass and I try to extract the nickel from this biomass. But to answer conventionally, all of this mass will be dried up, combusted completely. We're going to extract nickel by acid leaching. Mediation project allows that because it like Dr. Ratna said, it and the only thing that can be done here is that plant species can translocate from soil roots. So Alif, that is the answer. Yeah.
Okay. Uh, do you have an idea on the general yield over time? For example, if you leave the plant for 6 months, how much is the yield?
Okay. Your
Let's maybe your experience, Dr. Yong. Because the process in phytoremediation, I present earlier, that it's not a fast process, but it's a long-term process. Maybe it depends on the type of plants and the type of soil conditions. It's contaminated is just a surface or a deeper in soil. Maybe we can monitor by see maybe we get the biomass and analyze this in the lab. We can use as maybe accumulation analysis with AAS, I think, and we can see how milligram or how nanogram per kilogram dry weights in that some point, and we can monitor the next 2 months or the next 2 months again, and we can see the trend, how long the contaminants is already stored in the plant tissue. Maybe after we know the trend, we can determine how long we harvest the biomass to get the heavy metal from the plant's tissue. Maybe you agree with me, Dr. Yong?
Yeah, I totally agree with you. What Alif have asked earlier is quite deep, quite technical, worthy of a
Yes.
postgraduate student's level.
Ah.
And anyway, Dr. Ratna, we already reaches 10 o'clock in the morning. So I believe we have to end this Q&A session. But Alif, don't worry. If you're interested to pursue this area study, the good news is we UITM have a team in Sabah focusing on that one. So we very much welcome to join that doesn't mean that UNIVERSITAS Indonesia also may have their own team. So before I end this session today, I would like to thank all of the participants to joining this session. And before we end, I would like to encourage my student to consider the programs, international programs promoted by Universitas Indonesia. In fact, just 1 month ago, Universitas Indonesia has promoted the exchange program with UITM. So this is the result from our MOU signed with Universitas Indonesia and also the Faculty of Law, UITM. So just to recap, there are many more international programs between us. So for your students, if you want to join us, please do so. We do have a lot of programs that can welcome you. Exchange or internship, please find more information in UITM's and also Universitas Indonesia's website. So with that, once again, thank you, Dr. Ratna, for the insightful and informative talk on the topic of phytoremediation. If we have more time, I wish I could talk more specifically.
Yes.
But this is the time that we have. So thank you very much once again. And before we leave, perhaps we can all switch on the camera for one photo session.
Yes.
So please give me a second and let me adjust the view so that I can allow as many people as possible. Yep. So unfortunately, my screen can only allow up to like maximum 50 plus images. So students, switch on your camera and I will do a countdown for a photo. I mean, this is for reporting, Dr. Ratna, we have to do this.
Yes.
Okay, so you ready? Everyone, switch on your camera. I still haven't seen all of you with your camera. So as many as possible, especially for those who are holding a smartphone. So if you're holding a smartphone, there will be more space that you can cram up in the screen. Still not everybody switch on their camera. Anyway, I still have to. Okay, so I will do a countdown up to 3. So if you switch on your camera, so 1, 2, 3, cheese. Okay, hold on. Let me save this file. Hold on a second.
Dr. Yong, I will share my slide presentation to you later today.
That would be great.
Yeah, I will send via email maybe or WhatsApp.
Thank you very much. I will going to share the PowerPoint slides in with all the audience. So there you go. We have reached the end of our invited lecture. So thank you very much for joining us. So have a great day.
Yes, have a great day.
Thank you, everyone.
Bye-bye.
Goodbye.
Bye-bye. Goodbye. Thank you, Dr. Yong, Dr. Lily.